Hello, everybody, and welcome back. Good afternoon or good evening, depending on when you are watching this. This is going to be our last chapter for the A&P 1 lecture class. So we're going to do the special census today, which is chapter 17. We're going to try to keep this light because you guys have so many other things to study as is. So I'm going to try to make this as straightforward as possible. So, the special senses are referring to senses that we have that are, of course, specialized. So, olfaction, our gustatory sense, which refers to our sense of taste, vision, as well as hearing and equilibrium, those are going to be our special senses. So, olfaction is going to be the first one that we're going to mention. So, that's our sense of smell. Olfaction is referring to the sense of smell. This is brought in here by cranial nerve 10. It's going to be the nerve that brings this information in here. And of course, this is a chemical sense. What's important here when we talk about this being a chemical sense is that we have this olfactory epithelium that's actually covering what is known as the cribriform plate. That's going to be located in the nasal cavity. If we can think back to our lab class, the cribriform plate is going to be part of the ethmoid bone, and so the olfactory epithelium is going to be covering this cribriform plate, and in smell information, which is essentially different chemicals, are going to come in and get dissolved in the layer of mucus that's on the outer portion of the olfactory epithelium, and the olfactory epithelium has different receptors that are going to be able to take in that information and send it up to the brain. So you can see that the nose has quite a big many different types of receptors for smell of the olfactory epithelium. Excuse me, and so there's many different types. And again, this olfactory epithelium is going to be found up here in this region. So here is the nasal cavity right in here. You can see that the ethmoid bone is going to be found right here, and this is the cribriform plate. So the cribriform plate is found in this little region. The cribriform plate is these little holes going through it, and through each hole are going to be projecting these axons of different neurons. You can see these axons here projecting through the holes, and they're going to be synapsing on what are known as glomeruli, and the different glomeruli are going to be then synapsing with mitral cells, and those are going to lead back to the brain. And you can see the olfactory bulb right here is going to be our structure that we see on the brain as well. So you can see that these different neurons, these olfactory receptor cells, are going to have their dendrites here in this layer of mucus. These are going to be part of the olfactory epithelium, and these olfactory receptor cells are going to be sending that information up here to these glomeruli through the cribriform plate. And they're going to be synapsing there with the olfactory bulb, or specifically those glomeruli in the olfactory bulb. So those impulses are then going to travel along the olfactory tract back this way to the brain. So the primary olfactory area, which is going to be found in the temporal lobe, will be taking in this information. So again, these are bipolar neurons. This is one of the places we see bipolar neurons. You can see that there's little receptors here on the end of these cells, and that's going to send that information back to the brain. So that's that's pretty much what we want to know on olfaction. So the next sense is going to be gustation, or the sense of taste. So there are 5 primary tastes that we want to know. So sour, sweet, bitter, salty, and umami. So umami is kind of an interesting one. This is a meaty or savory flavor. We get this in a lot of nice chicken broths or something like that. So that's where we might taste umami. And you're probably a little bit more acquainted with the rest of these. So essentially with gustation, or our sense of taste, as we say, there's going to be many different taste buds that are found on the tongue. This here is a taste bud, and a taste bud is made up of many different cells that are sort of grouped together in a group, and they're going to give us our sensation of taste. So there's going to be roughly 10,000 taste buds found on the tongue, but they're also going to be found on the soft palate, the pharynx, and the epiglottis as well. So there are taste buds in many different areas, not just the tongue. And there's three main cells that we find in these taste buds. The most notable would be the gustatory receptor cells, which are going to give us the sensation of taste. The basal stem cells are going to be sort of repairing and taking the place and turning into other cells because they are stem cells. And then our supporting cells will be, you guessed it, supporting. So this is kind of a structure of a taste bud. This whole thing here is going to be a taste bud. And again, we have these different taste receptors that are going to be on the ends of our taste buds, and then this information is going to get brought back to the brain through that manner. So there's three cranial nerves that are going to help giving us our sensation of taste. So the first one here is the facial nerve. The facial nerve is going to be taking taste information from the anterior 2/3 of the tongue. So up here, you can actually see that this here is the facial nerve. This is a very good picture. This is the facial nerve, and that's taking in information here from the anterior 2/3 of the tongue. The glossopharyngeal nerve, on the other hand, is going to take taste information from the remaining 1/3 of the tongue. So back here, you can see that this is the glossopharyngeal nerve. So I do want you to know these three nerves here, and again, you can see these different nerves that are shown here in the different areas that they are taking information back to the brain with. So that's all we're going to touch on here with gustation. We're going to talk about vision. Vision is going to be a little bit longer for us. We have a little bit more to talk about with vision. So there's a few. We're going to start with the anatomy of the eye. So the first portion here is the cornea. This here is the cornea. The cornea is going to be this clear layer here found on the most outer layer of the eye. So the cornea's main function here is going to be to admit and refract light. Refraction just means to bend light, and so light's going to come in here through the cornea. That's the main idea. This outer portion, this white portion of our eye, you can see this portion here. This is the white part of the eye. This is known as the sclera. And the sclera's main function here is going to be to provide structural support as well as providing shape to protect the inner parts of the eye. So this is a very protective layer is here, the sclera. That's the white part of the eye. The next portion we're going to mention here is the iris. That's going to be the colored portion of the eye that we see here. And the iris's function is very, very interesting. I'd say this is probably one of my favorite parts, which means that I might ask a test question about the iris, hint, hint. So the iris is going to be regulating the amount of light that goes into the eye. And what's happening here, the way that this is going to work here, is the iris is actually going to open or close, or we say dilate or constrict, based on a set of different stimuli. So you can see that the little hole here found between the iris is known as the pupil. So the pupil is the little dark area here in the middle, and the iris is the colored portion around the side. Next up, we have the lens, which is going to be found right here. That's this big structure here in the middle. The lens's main function is going to be to refract light. And so you might remember that I said the cornea refracts light, or reflects light. But the lens is really going to be the main player in refracting light. So that's the main function there of the lens. Now the lens is going to be connected to what is known as the ciliary body, which is this structure right here, through these fibers known as zonular fibers. Zonular fibers. Those are going to be found here. So to kind of review that, the lens is attached to zonular fibers, and the zonular fibers are going to be attached to the ciliary body. So the ciliary body's main function is going to be to alter the shape of the lens. You can see that this has a muscle in it here in the ciliary body. And the muscle is going to change the shape. As the muscle contracts, it's going to pull on the zonular fibers, and that's going to change the shape of the lens. And that's going to allow us to see at different levels of depth. The next layer I want to mention here is the choroid. The choroid layer is going to be a layer of the eye that is essentially functioning to bring blood flow to the eye. So this is providing a blood supply to the eye because there are lots of blood vessels here found in the choroid. And this is also a very pigmented layer which helps us to absorb excess light that has gone into the eye. And the last structure I want to mention here, or sort of the last two structures here, is what is known as the retina. The retina is this yellowish layer. You can see the yellow retina here. This is going to be going along about 3/4 of the back of the eye. And the main function of the retina is to receive and convert light to different potentials and nerve impulses. So this retina is going to be the place where we find rods and cones. So that's going to be the main function of the retina. Is this is actually going to convert light signals into something that we understand. And so in the back of the retina, you can see that we have this here is the optic nerve. We have this area where all this information is going to come here and leave the eye through the optic nerve. So this is cranial nerve number 2. So the optic nerve is going to be sort of entering and connecting with the retina at this spot, which is known as the optic disc. And what's quite interesting is the optic disc is actually what is known as the blind spot. So we actually have this area where we don't have any photoreceptors. Photoreceptors is a fancy word for rods and cones. So we don't have any photoreceptors in this place, so we actually can't see anything with this spot at the optic disc. So the idea here is light's going to pass through the cornea. It's going to go through the pupil, which is that little hole here that's going to be found right here. Again, here's the iris. That light information then is going to go through the lens and get bent, and the goal is to get that image focused onto the retina. And that's how we have good vision, is by focusing a picture onto the retina. So once information gets back to the back of the retina, this visual information is going to cross at the optic chiasm. Remember that the two optic nerves are going to cross at the optic chiasm, and then the information from each eye is going to go to the opposite side of the brain. So the information taken back from the right optic nerve is going to go to the left hemisphere, or to the left occipital lobe of the brain. And the opposite is the exact same. So the retina is going to be lining most of the back of the eye. This is what it's going to look like here. The macula lutea is this area of highest visual acuity. You can see that down here. That's going to be found in the center of the retina. So the macula lutea, this is the area of highest visual acuity. So this is where we can really see structures very, very clearly. So you can see that right here is the optic disc. This is going to be the place where the optic nerve is entering the eye. This is where we have the blind spot. So that's what the optic disc is going to be. The optic disc's main function here. So you can see that this is going to be a picture that our physicians are trying to look at in us, and they want to be able to see the retina and make sure that we have healthy eyes when you go to the doctor's office. So the retina is going to contain those photoreceptors, like I mentioned. Those are rods and cones. So what I'd like you to know about rods is rods allow us to see in dim light. And what I'd like you to know about cones is that cones are going to produce color vision. So C for color. Cones produce color vision. Rods allow us to see in dim light. Rods don't have any color vision. Cones are the ones that give us our color vision. So essentially, these rods and cones, these are different photoreceptors, different types of cells that we have in the eye. They're going to send their information back down the optic nerve into the back of the brain. So this is some review here. I'm not gonna touch on all these, but this is some review of what I've covered here on the eye. So I'll let you guys go through this information at your own leisure. So again, in a normal eye, which is called an emmetropic eye, this is a normal eye, the light is going to come in here through the cornea. It's going to go through what is known as the anterior chamber, which is this space here, and it's going to get reflected here by the lens onto the retina. And this is where these light rays come in and focus. That's known as the focal point. So we want the focal point to be directly on the retina, and this will give us a very clear image. So when we are looking at an object versus, that's close up versus an object that's far up, you can see that the ciliary muscle is going to contract, and that's going to sort of change the shape here of the lens. It's going to be more spherical here. When the ciliary muscle is relaxed, the lens is going to be more flat, and you can see that makes a difference here in the difference of the rays that are coming in. So you can see that these rays that come in here from a close object are going to be different than they are whenever they come in with a distant object. So that's what's going to be happening here with the difference between seeing up close versus seeing far away, is that the lens is changing its shape to give us the ability to see the difference between a close object versus a far object. So when talking about some pathology here of vision, myopia, which is also known as nearsightedness, is referring to a condition where our patient cannot see far away. They can only see near. That's how I remember that. Near is in the word, so they can only see near. So what's happening here is the eyeball is longer than it should be. You can see that this here is the eyeball. It's the entire eyeball, and this is the way it's supposed to be. This is the way the typical shape of the eyeball here. Some of us that some of you might actually be myopic. We're going to learn farsightedness as well, so we're not going to leave you guys out either. So light rays are going to come in here, and they're going to converge on a place here that is before the retina. So the focal point's not going to be on the retina, and that gives us a blurry visual picture. So typically, this is going to be treated with a concave lens. This is going to help reflect that light even more, so that we can bring the light back to the retina. So again, what I want you to know here is that nearsightedness is referred to as myopia, and that this focal point's before the retina, not actually on the retina. So this is an elongation here of the eye. In cases of hyperopia, which is known as farsightedness, this is where our patients can only see far. And so this is the opposite, where the eyeball is going to be shorter than what it's supposed to be. You can see that this dotted line here is showing us where we are supposed to be. And you can see that this eye here is not quite as long as it's supposed to be. So farsighted individuals, they can see very clearly far away, but they have great difficulty seeing objects that are up close. So again, this is called hyperopia, or these patients would be hyperopic, and a convex lens is going to be typically used to treat this condition. Astigmatism, on the other hand, is referring to a condition where there is an irregularity in the curvature of the eye. So what this leaves us with is multiple focal points. That's astigmatism, irregularities in the cornea. Or you can also have them irregularities in the lens, or both. But we'll look at the cornea, and we can think about the cornea producing multiple focal points. So this is going to give us two different focal points, and that's going to, you can see the effects here of astigmatism. You can see this blurriness that's caused here by astigmatism. So hearing and equilibrium are going to be our final senses that we're going to talk about tonight. So there's three major regions of the ear: the external ear, the middle ear, and the internal ear. So there's three regions, and we're going to talk about each one of these regions and what's contained in each. So the external ear is referring to or it's containing this structure known as the auricle, which can also be called the pinna, and that's just the outer ear. That's just the part of the ear that you can actually grab and touch. I'm touching it on myself right now. You can kind of feel it. It's made up of cartilage, and it's sort of shaped like a disc a little bit. That's the outer portion. I'll show you what that looks like. The external auditory canal is referring to the canal for which sound information goes into the ear. So where you stick the Q-tip, this is the external auditory canal. The other portion here of the outer ear, the external ear, is the tympanic membrane, which is also known as the eardrum. So the auricle is going to capture sound just like a satellite disc would capture signals. The external auditory canal is going to take those signals to the eardrum. And so if you can think back to chapter 5, we've talked about ceruminous glands, which are going to be secreting cerumen, which is a fancy word for earwax. That's going to protect the canal and the eardrum as well. So this is a picture here of the external ear. You can see that this is the auricle, or the pinna. This is the ear, as we know it, the ear. This region in here is the external auditory meatus, or the external auditory canal. You can see that this is the eardrum, which is also known as the tympanic membrane. So the middle ear, these are going to contain the three auditory ossicles. Now I know that if you had my lab, actually, you wouldn't have needed to have this in my lab. We actually did this in my lecture in, I think it was chapter 7 in the axial skeleton. These are going to contain, the middle ears containing the three auditory ossicles. So those are the malleus, the incus, and the stapes, and these contact each other in sequence. So here's the malleus. You can see that the malleus is actually going to contact the tympanic membrane here, and when vibrations come in at the tympanic membrane, then it's going to vibrate the tympanic membrane. That's going to push on the malleus bone. The malleus is going to push on the incus bone, which is our second bone here, and then the incus bone is going to push on the stapes bone, which is our third bone, and then that pushes on what is known as the oval window, and that translates that sound information into the inner ear. So the last thing I want to mention here is we do have here in the middle ear what is known as the eustachian tube. It's labeled here the auditory tube. It's called the pharyngotympanic tube here, but I like eustachian tube, so it'll be eustachian tube on my test. So the eustachian tube is functioning to equalize pressure between the internal and external environment. So if this was cut off here, you can think that when the pressure changes outside, that's going to pull the tympanic membrane, depending on the way the pressure changed, either in or out, and that could be potentially very damaging here. It could affect our hearing. So the eustachian tube is functioning to equalize pressure between the outside environment and the middle ear. And this is, again, why, whenever you fly in a plane or something like that, and you go way up high, or, drive in the mountains or something like that, the pressures change, and that's going to possibly affect your middle ear. So again here, we can see these different bones here found in the middle ear, and here is the eustachian tube that's going to lead to the nasopharynx. That's going to take us into the inner ear, which is also known as the labyrinth, because it's very, very there's a lot of little pathways that are going to be found here in the inner ear. So the main structure we see here is the cochlea. The cochlea is referring to this spirally looking structure here. It looks like it'd be on the back of a snail, or it looks like some type of French horn or something like that. But the cochlea is going to be the structure that translates vibration. So there's going to be fluid that's found inside of this structure here. This is what the inner ear looks like, this bony structure right here. So there's going to be fluid that's filling in these different little pathways in here. And so the main idea is when the stapes hits on the oval window, which is going to be found right here. This here's the stapes pushing on the oval window. That's going to transmit sound information here into the cochlea, and that's going to vibrate this fluid in here, and that's going to cause a cascade of events, and that's going to take information, back to the brain, essentially, in so many words. So that's the main function of the cochlea, is for hearing information. There's also the semicircular canals, which are going to be these canals right here. These are going to function in equilibrium. So these are, I find it quite interesting that these canals here are going to actually be positioned at about 90 degrees from each other, because this allows us to feel motion in all directions. So again, when the fluid here is sort of rotating or flowing through these different canals, that gives us the sensation of motion. So that's going to be localized here to the semicircular canals, and again, you can see those canals right here. There's also this bulbous structure right here that's going to be found between the cochlea and the semicircular canals, and this is going to be made up of what is called the utricle and the saccule, and we will touch on those in just a minute. So in terms of equilibrium, when we talk about equilibrium, there's two different types. There's static equilibrium and dynamic equilibrium. Static is when we are stationary or not moving, so the maintenance of our position when we're just considering the force of gravity. So that's our static equilibrium, when we're standing still or sitting still or something like that. The dynamic equilibrium is referring to the maintenance of our body's position when we're dealing with the movement. So the way that you might be in a car when you are accelerating or something of the sort. So that's our dynamic equilibrium. And the vestibular apparatus refers to the different structures that give us our sense of equilibrium. So, for example, that saccule and utricle that I mentioned, those are what are known as otolithic organs, and I'm going to explain that in a bit, as well as the semicircular canal. So all three of, or actually, excuse me, yes, I said that right. All three of these organs here, these different structures, are going to be part of the vestibular apparatus. So these are the three things that make up the vestibular apparatus. So we said that these made up the otolithic organs. We have other structures that are found in the inner ear known as otoliths. They're just calcium carbonate crystals. They're going to be forming a layer over the otolithic membrane, which is going to be, I'm going to show you that here. So this here is going to be the otolithic membrane. That's this structure right here. And we have these different cells known as hair cells. And we're not going to learn much about hair cells, but these are going to be cells that are projecting what are known as cilia. So we have cilia. We've talked about cilia before. These are projecting cilia. And so the idea here is we have these little otoliths that are going to be sort of, they're going to be providing a weight to this otolithic membrane. And you can see that when this guy here has his head upright, you can see that this is going to be the structure here of our otolithic membrane. And when he puts his head forward, he tilts his head forward, you can see that this is going to, these otoliths are going to push this way, and that's going to warp the different hair cells or the cilia on the hair cells, and that gives us a sense of motion and movement. So in summary here, again, the auricle is going to collect our sound waves. The external auditory canal is going to take sound to the eardrum. The eardrum is going to capture those vibrations, and that's going to transfer the stimuli to the malleus. The auditory ossicles are the malleus, incus, and stapes. Those are going to take the information from the vibration information from the tympanic membrane to the oval window. And then, of course, the eustachian tube is going to equalize air pressure on both sides of the tympanic membrane in the middle ear. Our last summary slide here is the cochlea. This is going to be containing a series of fluids in different channels, and this is going to be essentially giving us our sense of hearing. So this is bringing different types of hearing information back to the vestibulocochlear nerve. The vestibular apparatus is going to include the semicircular canals, the utricle, and the saccule, which are going to send the information from the balance information back to the vestibulocochlear nerve as well. Remember, the vestibulocochlear nerve is cranial nerve 8, and this is going to deal with our balance and hearing information. The semicircular canals, this should say canals, semicircular canals, is going to detect different types of acceleration based on the positioning of those canals in many different orientations. The utricle, one thing I didn't mention, is going to detect linear acceleration or deceleration. So that's going to be detecting acceleration on a linear path, so essentially in a horizontal direction. The saccule, on the other hand, is going to be detecting acceleration in a vertical direction. So hopefully that wasn't too dense for you. I tried to get through that quickly so you didn't have to spend too much time on Chapter 17. I just wanted to touch the general idea with these special senses. Other than that, I hope you guys have a fabulous rest of your night. Good luck studying, and please feel free to reach out to me if you need anything at all.